Welcome back to General Biology 1 lecture. For this video, it will focus on the parts and functions of cell membrane and its role in the acquisition and removal of substances within the cell. What is a cell membrane? Cell membrane, also called plasma membrane, is a thin membrane that surrounds every living cell, delimiting the cell from the environment around it. Enclosed by this cell membrane are the cell's constituents, often large, water-soluble, highly charged molecules such as proteins, nucleic acids, carbohydrates, and substances involved in the cellular metabolism. Outside the cell, in the surrounding water-based environment, are ions, acids, as well as nutrients that the cell must absorb in order to live and grow. We need to take note that cell membranes protect and organize the cells. All cells have an outer plasma membrane that regulates not only what enters the cell, but also how much of any given substances come in. What do membranes do? Cell membranes serve as barriers and gatekeepers. They are semi-permeable, which means that some molecules can diffuse across the lipid bilayer, but others cannot. Small hydrophobic molecules and gases, like oxygen and carbon dioxide, cross membranes rapidly. Some polar molecules, such as water and ethanol, can also pass through membranes but in a slower rate. On the other hand, cell membranes restrict diffusion of highly charged molecules such as ions and large molecules such as sugars and amino acids. The passage of these molecules relies on specific transport proteins embedded in the membrane. Cell membrane is also selectively permeable, wherein it regulates the substances that will enter and exit the cell, meaning allowing only certain molecules to cross and relaying messages via a chain of molecular events. And lastly, cell membrane is composed of a mixture of lipids, carbohydrates, and proteins. In short, if the cell is represented by a castle, the plasma membrane or cell membrane is the wall that provides structure for the buildings inside the wall, regulates which people live and enter the castle, and conveys messages to and from neighboring castles. Just as a hole in the wall can be a disaster for the castle, a rupture in the plasma membrane causes the cell to die. The most accepted model for cell membrane is the fluid mosaic model, which was proposed by S.J. Singer and Garth L. Nicholson in 1972. The fluid mosaic model describes the structure of the plasma membrane as a mosaic of components, including phospholipids, cholesterol, proteins, and carbohydrates that gives the membrane a fluid character. The fluid there means that the components of the cell membrane are constantly moving, and the mosaic means that the cell membrane is made up of different substances with different functions. Let's proceed by discussing the different components of the cell membrane. First, we have the lipids. And under lipids, we have phospholipids, cholesterol, and glycolipids. Take note that almost 90% of the entire cell membrane structure is made up of different types of lipids. The main fabric of the membrane is composed of amphipathic or dual-loving phospholipid bilayer molecules. The hydrophilic or water-loving areas of these molecules are in contact with the aqueous fluid both inside and outside the cell, while the hydrophobic or the water-hating molecules tend to be non-polar. So the hydrophilic head is made up of phosphate group wherein it has affinity with water, whereas the hydrophobic tail made up of fatty acids does not. On the tail of the phospholipid is a kink. The kink is due to the double bond in the unsaturated fatty acid. The kink prevents the tight packing of the phospholipid, hence its fluid movement. A phospholipid can move in the bilayer in several ways. A phospholipid can move on its axis through rotation. It can also move sideward through lateral diffusion. It can also move from the inner layer to the outer layer or vice versa through transversal diffusion or flip-flop diffusion. Take note that transversal diffusion occurs the least. The next type of lipids located in a cell membrane is a cholesterol, wherein they are found between the phospholipids that act as a fluidity buffer. Cholesterol is very important in the membrane as it helps to maintain cell membrane stability and fluidity at varying temperatures. At low temperature, cholesterol reduces their packing, which means that at low temperature, when the rate of movement is lowest, a fluid phase is maintained. While at high temperature, cholesterol helps to stop the formation of crystalline structures, therefore making the membrane less fluid. The cholesterol acts as a fluidity buffer. During warm temperature, it makes the movement of phospholipids limited, making them less fluid. During low temperature, it prevents the close packing of the phospholipids, thus increasing their fluidity. So phospholipids and cholesterol are important in maintaining the fluidity of the cell membrane. Failure of these two to function may have detrimental effects to the cell. For example, let's say that a cell is exposed to high temperature and the phospholipids and cholesterols cannot keep the normal fluidity of the cell membrane. When this happens, an electrolyte imbalance may result in the cell, which may eventually cause the cell to swell or cause certain organelles to have impaired functions. The second major component of cell membrane, we have the membrane proteins. A typical cell membrane consists of around 60% protein. There is such a high proportion of protein because they are so vital in almost every process within a cell. As part of the cell membrane, proteins can either be deeply embedded within the bilayer, which is called the integral protein as presented in the PowerPoint presentation, or be associated with the surface of the cell membrane, which is called the peripheral protein. The integral proteins involved in facilitated transport are collectively referred to as transport proteins, and they function as either channels for the material represented by the left picture or carriers represented by the right picture. In both cases, they are transmembrane proteins. So channels are specific for the substance that is being transported. They are either open at all times or they are gated. Take note that the gate controls the opening of the channel. And one good example of channel protein is aquaporin, wherein it allows water to pass through the membrane at a very high rate. Another type of protein embedded in the plasma membrane is a carrier protein, wherein this protein binds a substance and in doing so triggers a change of its own shape, moving the bound molecule from the outside of the cell to its interior depending on the gradient involved. Take note that carrier proteins are typically specific for a single substance, and this selectively adds to the overall selectivity of the plasma membrane. Carbohydrates are the third major component of plasma membranes. They are always found on the exterior surface of the cells and are bound either to proteins forming glycoproteins and to lipids forming glycolipids. Along with peripheral proteins, carbohydrates form specialized sites on the cell surface that allow cells to recognize each other. This recognition function is very important to cells as it allows the immune system to differentiate between body cells called self and foreign cells or tissues called non-self. These carbohydrates on the exterior surface of the cell, the carbohydrate components of both glycoproteins and glycolipids, are collectively referred to as the glycocalyx, meaning sugar coating. So glycocalyx is highly hydrophilic and attracts large amount of water to the surface of the cell. This aids in the interaction of the cell with its watery environment and in the cell's ability to obtain substances dissolved in water. Thank you for listening. I hope you were able to learn something about cell membrane.